All right, it's 3 o'clock, so we will go ahead and get started. Welcome everyone to the 2024 training webinar for spiny surveillance. We're excited to have you join us today. I'm joined by Don Brandstratter and Val Brady, our two spiny water flea experts. And also on the line is Michelle Isaacson, the program coordinator for the AIS Detectors program. And I'm an extension educator focusing on aquatic invasive species at the University of Minnesota as well. So I'll be hosting today and have a couple of spots where I'll join Don in chatting. And Val is here as well to chime in and help with all things spiny. A couple of housekeeping items for the webinar. Everyone is muted, but we have a great tool that we called Slido that we're using for question and answers. So you'll be able to go to slido.com and you can enter any questions that you have there, and you can also upvote questions. So the most pressing questions always get answered first. If you struggle with it, don't worry. We've got you covered in chat. Michelle will help transfer things over as needed. We're also recording the webinar, so you can watch for that recording. It takes us a little bit to transcribe it, but we will post it onto our YouTube channel later, and we'll send out a notice when that's ready, so you can review anything or share with folks who weren't able to make it. Don is going to be kicking things off, so I think I'm going to turn it over to Don, and let me stop my share so that you can share yours.
That sounds good. Thank you, Megan, for that nice introduction. I'll get my slides going here and then I'll. Okay, everybody see that? Good. All right. Thanks for being here, everybody. It's a pleasure to have this opportunity to help lead or sort of co-lead this webinar. And what we're going to try to do here today is talk to this topic here, spiny water flea surveillance, and tell you, kind of backfill that, tell you why we're doing this, why it's important, and then spend some time talking about how you can become involved and get engaged and help with this effort. So the title here is spiny water flea surveillance, sort of keeping eyes on the water, and we're trying to engage as many people as possible to help us in this effort through this volunteer-based program. So why is spiny water flea what is spiny water flea surveillance monitoring? It is more technically sort of this idea of strategically implementing and with a lot of coordination surveys with a level of effort that have been evaluated to help us understand essentially presence absence. Is spiny water flea in this lake? Are they not in this lake? And that in fact is critical information that's going to help us update current distribution maps in Minnesota. And by knowing current distribution maps, especially where new identifications might be made, that can help us mobilize early efforts for containment. This sort of naturally, logically is a next step following a pretty big campaign called Stop Spiny that had a lot of hands involved, a lot of resources involved in helping expose the threats of spiny water flea, understand the threats of spiny water flea to lake ecosystems in Minnesota, and how we can help deal with some of those threats and continue to prevent spread. The Spiny campaign has all sorts of resources. It's housed online and maintained through the Minnesota Aquatic Invasive Species Research Center. The surveillance monitoring piece of it is an outreach of that, in fact, and it's relatively new. We first piloted this in 2022. We had a nice buy-in of volunteers then. We continued the program in 2023. I'll show you a little bit of data later in terms of how many participants we've had and what they've done. And so this is into 2024, and we're hoping to continue to do nothing but continue to grow the program through engaging more citizenry to do this. There's been lots of hands in this effort. I'm in the upper left there. Val Val Brady is in the upper middle there. She's on the call today as well. Both of us are associated with the University of Minnesota Duluth. I'm in the biology department. Val is in the Natural Resources Research Institute. Formerly at MAISRC is Meg Durr, and she was a huge critical part of mobilizing this and really assembling the resources that are really the heart and soul of this surveillance monitoring effort. And now we're happy to have Megan Weber and Michelle Isaacson as part of the team to help continue the effort of maintaining and growing this program in Minnesota. So why conduct surveillance for spiny water flea? Well, despite the fact that spiny water flea, and I'll provide some more data in just a second on this, they've been in the inland lakes of Minnesota since 1990. But during that time, their spread across the landscape can probably still be considered quite limited. There's only about 36 lakes currently in Minnesota, plus or add or subtract a few that have established populations of spiny water flea. And all of those lakes, you can kind of see from this map here, are located generally in the Northeast region, in the Arrowhead region of Minnesota, the land of 10,000 plus lakes. There's thousands of more lakes in Minnesota that have suitable habitat for supporting spiny water flea. And we'd like to do our best to prevent any of those from being invaded. We have no formal monitoring currently at hand there, but there's plenty of people on the lakes all the time, living on lakes and moving between lakes, who can help participate in the effort of continually mapping presence and absence of spiny water flea. And so the surveillance effort is really important. This is just sort of an interesting piece of data that shows you the timeline or the track of numbers of lakes in Minnesota that have been successfully invaded, where a spiny water flea population has established. So I indicated that it was around 1990 when the first inland lake, which was Island Lake Reservoir near Duluth, had a confirmation of establishment. And then we went for like 10 or 15 years, and then we found up on the Gunflint Trail, Lake Saginaw in the early 2000s, and then climbed up. Most recently, Snowbank Lake in northern Minnesota in the Ely region had a confirmation, but the line is flattening out. We're sort of reaching a plateau, perhaps, if you will. There might be a number of different reasons for this, but we're hopeful that a lot of that might actually have to do with sort of more conscientious boating and movement of human traffic from one lake to the next, and taking care of cleaning equipment as people move. One of the interesting things, though, about spiny water flea is that once they invade a lake and do so successfully, they grow to relatively large numbers. So amongst the lakes that are invaded in Minnesota, those lakes support anywhere between 55 and 50 individuals per cubic meter of water in the summertime. So the lakes that are invaded are supporting populations on the sizes of millions to tens of millions of individuals. One of the other things that we know through MAISRC-supported research and St. Louis County-supported research is that angling equipment that is trafficked by boaters between lakes is highly susceptible to hitchhiking by spiny water flea. So recreational gear, especially fishing lines, maybe downrigger cables, live wells, are very susceptible to ensnarement of spiny water flea. And so there's still plenty of opportunity, right, on the landscape, given recreational boating to move spiny water flea from one lake to the next. That schematic on the right there shows sort of a visual of the connectedness of Lake Mille Lacs, which is one of our 36 invaded lakes, and its connections to lakes throughout the state vis-a-vis recreational traffic. The other thing about spiny water flea is when they're clumped up on equipment, as you see there in that center image, They're curiously different and unique, and one's eyes might gaze at them and wonder what that is. But as individuals ensnared on a line or on some other piece of equipment, they can be really difficult to see. And so hitchhiking easily can occur easily from one lake to the next when eyes aren't used and effort isn't used to clean equipment. And that idea that fishing line in particular is highly susceptible to ensnarement was the basis for the title of a publication that Val Brady and I and others put out a few years ago, where we said line snag spines. That was the headliner of that research effort. So we know a number of other things about spiny water flea as well that can help motivate us to invest resources in surveillance monitoring. We know that spiny water flea has a high impact on the food webs, and we'll talk in just a minute about what some of those specific impacts are on food and energy transfer in aquatic food webs. Like most other aquatic invasives, we know of no control methods once introduced to a body of water, and so prevention or containment is our only real way to fight back. And knowledge about where they are, okay, clearly is going to help us focus efforts in identifying lakes as high risk or not. And so knowing even if efforts on surveillance catch only a single individual in a lake, it's almost certain that that spiny water flea has been there probably for a year or more. So even a single or two individuals can help us identify a lake as invaded. So let's talk a little bit now about what the deal is with respect to ecosystem disruption and modification upon the arrival of something of spiny water flea. So this schematic here demonstrates, those arrows demonstrate the way in which energy or nutrients move from one trophic level, if you will, or one level of an aquatic food chain to another. You can sort of see the arrows going from the primary producers or the phytoplankton in the upper left to the grazers or the zooplankton, which are the primary food source for a variety of different fishes, some throughout their entire life, like zooplanktivorous fishes, perch and bluegill, and then all the way up to fish-eating fish or piscivores. So what happens when spiny water flea arrives in an ecosystem? We know a lot of the outcomes now through a variety of directed research in the laboratory, in the field, and a lot of unfortunate experiences of spiny water flea entering into food webs of lakes and looking at sort of before and after changes. So spiny water flea pictured there at the end of that red arrow is a carnivore. So it feeds essentially at the same trophic level as do things like perch and bluegill. So spiny water flea feeds on the native zooplankton, and they are fast growing. And as I've already mentioned, their densities in lakes can be very high, 5 to 50 individuals per cubic meter. And they can very easily drain off a lot of the energy and nutrients in those zooplankton that would otherwise have flowed to zooplanktivorous fish. And that's one major effect. One thing that we know for sure, it's been repeated over and over again, is that when spiny water flea arrives in an uninvaded ecosystem, they suppress native zooplankton, not only the numbers, but also the biodiversity. One of the unique things about spiny water flea among the freshwater zooplankton is they have this long caudal appendage. It's sort of their namesake appendage, the spiny water flea. And that can prevent small fish that are smaller than a few inches in size, from successfully consuming them. And so they, in that fashion, they don't represent an alternative food source for fish, but for small fish anyway, but really an energy and a nutrient dead end. And as a consequence, some of the more recent research is showing that zooplanktivorous fish and fish that rely on zooplanktivorous fish, things like walleye, may grow slower. And in fact, there's evidence in a number of lakes in Minnesota that they do grow slower. That sort of research took longer to get at, just because of the long-term data sets that are needed to effectively evaluate and check against other effects that could be occurring. Thirdly, I'll just say that because native zooplankton and those which are grazers, so feeding on phytoplankton, their numbers are suppressed. We have seen increased murkiness or decreased clarity in lake water where spiny water flea have invaded. And so their impacts are numerous, and they span up and down the aquatic food chain. Here's an image that one of my graduate students took of a spine, circled there by the red circle. A spine of a spiny water flea had punctured the gut lining of a walleye that was harvested from Island Lake Reservoir. So there's some internal health damages associated with the consumption of spiny water flea, and what the full effect of these are, we're still trying to understand and document that. Okay, so again, why conduct surveillance for the spiny water flea? Well, kind of because we can, right? And I'll just sort of give away the ghost here that we haven't, through this surveillance program, we haven't found any new invasions yet. All of the efforts have yielded 0 detections of spiny water flea. But in the world of aquatic invasives, 0 is our favorite number. We love to see zeros. It means that there's no new lakes that have been invaded, and you can't document a 0 or a no detection until you actually go out there and put in the effort to scan the water and to look at those samples and to really, with some degree of confidence, conclude that there aren't any individuals there at this particular time. And again, so we can do it. There's no laboratory test needed. We just need to go out and sample the water. The amount of the types of equipment and the variety of equipment that you need to do this is relatively basic, save for a couple items like the net, for example, and we'll talk about that in just a moment. And again, so these research-based guidelines can greatly enhance efficacy of survey methods. One of the things to consider and we've been asked this question, well, when can I and when should I do my surveys? Can I do them in the winter when I'm out there, ice fishing, or do I have to do them in the summer? What does this look like for spiny water flea? And it really has to occur in the summer during the open water season, and most preferably between June and October. And the reasons for that is that this organism has a really interesting life cycle where during the summer, during the open water, June to October, they exist in large numbers out in the open water of lakes. They sort of move from the surface to the bottom day and night, but they're out there in the open waters of the lake in this identifiable morphological form, this free-swimming planktonic form, shown there in the upper left. Come fall, though, and then going into the winter, they sort of exit, if you will, from the water column of a lake. They sort of, in some ways, operate like an annual plant, where in the fall, the females produce males. They mate with the males, and they make these eggs, these resting eggs, we call them. And there's a female in the upper right of this image, and she's carrying a batch of these golden spherical resting eggs. And then down the bottom, there's a couple other resting eggs sitting there. It's an image that I took a number of years ago. Those eggs sink to the bottom of the lake, and they just overwinter there. And so, for all intents and purposes, they're invisible to any collection methods during the course of the winter. All spiny water flea. So every population, even in a fully established lake like Mille Lacs or Island Lake, where they've been for 30 plus or so years there, every winter, the entire population commits to these seeds that go down into the sediments of the lake, and then the next summer they come back. But they're there for the open water part of the season, June to October. So there's plenty of time to detect them. And the detection is best done with nets that look like this. This is our research team out there doing some evaluations of ambient density. You can see a couple nets hanging there. We'll talk a little bit more about the ideal net in just a minute. So they're most easily detected, obviously, as I've spoken to, in the open water season, and typically during the day and night. The densities tend to be higher at night, though, than during the day. I don't want you to get too blown away by the multi-panels here, but each of these little panels shows an estimate of the density of spiny water flea that we measured at different depths in the water column. So if you look in the upper left there, the surface is at the top, that's 0 meters, and then 2, 4, 6, and all the way down to 12 meters. And then the little suns above and the moons above show our evaluations during the day and night. And you can just kind of see that at night, at the same location in the same lake, 2 or 3 or 4 times as many spiny water flea are available for collection. And that's because those animals, even during the summer, tend to go down to the bottom during the day and to come up at night, probably themselves chasing food and avoiding at least the bigger fish. So we suggest that if these surveys can be done either in the early morning hours or in the evening, as conditions and safety allow, the opportunity for detection is likely to go up. The other thing that we encourage a lot of our volunteers to do is to go out on this, whatever lake they're on or 2 lakes that they want to participate in with the survey, to go out multiple times during the course of the summer. Because one of the things that we know definitively about this organism is that its population densities during the summertime go up and go down. They go up and they go down. And so this is a time series from a lake around Duluth back in 2002, and you can see that this single population documented at a single location in the lake, those densities waxed and they waned and reached 5 or 6 peaks and valleys. And so going out 1 week and finding nothing really might not predict what you would find in that lake 3 or 4 weeks later. So multiple tries in the same lake might be needed to document presence. All right, so part of this surveillance is part of what we've done. The resources that we've put together include some informational videos, and I'm just going to show you one of these videos that demonstrates using a plankton net to collect spiny water flea. So we'll get this started here.
Hi, everybody. My name is Don Brandstrader, and I'm going to show you today how to use a plankton net to collect Vithotrephes, otherwise known as spiny water flea. So we're out here on Island Lake Reservoir in Duluth, Minnesota, and this is a lake that's known to have abundant population of spiny water flea. It is a relatively windy day, and so we anchor, and that's really important to anchor if there's really any wind whatsoever, to make sure your plankton net gets all the way to the bottom of the lake. We're in a location that's about 13 meters deep, and so I'm going to deploy this net to 12 meters and bring it to the surface. This is the net itself. It has a steel bridle, and it has an opening or a mouth diameter of about 1/2 meter, and the entire net is 1.5 meters long, and it is outfitted with Nitex mesh that has holes or apertures that are 500 microns in diameter. This is sort of an ideal net for collecting spiny water flea, but other variations on this theme will work as well. Down here at this end is the collecting bucket, sometimes called the cod end. And one useful thing that is important is to have some weight associated with that. So here are some 1-ounce, 2-ounce fishing weights that we coupled to the cod end with a zip tie, and that will help allow the net to go down vertically in the water when you deploy it. Okay, so we're going to put it into the water now, and we're going to make sure that the collecting bucket or cod end is submerged fully and full with water, so it deploys vertically. Then we're going to slowly lower the net. That'll ensure it stays vertical in the water column. Like I said, we're in about 13 meters of water. I don't want to go all the way to the bottom necessarily, because I don't want to pull up a lot of sediments. You can if you know that the bottom substrate is relatively rocky and inorganic, but I want to stay off of the bottom a little bit here. That's 10, and then that's 11, and that's 12 meters. When I've reached my destination, maximum depth that I want to deploy, I'll let it sit there for a couple seconds. Then I'll retrieve it, and it's just a relatively simple hand-over-hand retrieve, about a meter a second. You don't want to stop retrieving when you're pulling the plankton net, because you don't want any animals that are caught to sort of backwash out of there. We're at the surface now, and I'm just going to lift this net out of the water. Now, one of the things about spiny waterfly is that long tail spine tends to stick in and snare to some of the mesh. And to help encourage or discourage that from happening, once you get your net to the surface, a nice way to do a rinse is to do a dip, maybe 9/10 of the way down. You don't want to put it beneath the lifting ring and lift it back up just once. Okay, so that we've made our collection, and that's all for this video. In the next video, I'm going to dispense the contents of that into a sorting tray and show you how to identify spiny waterfly. We'll see you in the next video.
All right, so yeah, just to back up for a second, right? So we made.
My name is Don
Let me stop this. We ended up making, I think I made 3 videos, and then we had a 4th video that was something called lookalikes that was intended to help you all and anyone that looks at these videos distinguish between spiny waterfly and other native zooplankton that might be confused with spiny waterfly. So those are all available on the MAISRC Stop Spiny campaign website. Whoops, let's see if I can get this going again. All right, so getting started. Here, this is a list of equipment that would be sort of the ideal setup to begin to engage in this surveillance monitoring. The most likely piece of equipment that you don't have, but that you need, is going to be a zooplankton net and attached to a rope. That's pretty easy. There's a variety of different types of nets out there that could be adapted for this. The ideal net has mesh with holes in it that are 0.5 millimeter in diameter or 500 microns, and is a net that has an opening of about a 0.5 meter or so. And the only reason why this is ideal is because this sort of a net is going to exclude or not collect an awful lot of the phytoplankton and even some of the small zooplankton that's out there that might otherwise clog the net. But it's inconsequential to a survey of spiny waterfly. These organisms are big enough that they can be collected with nets that are going to exclude a lot of the organisms and debris and whatnot in the water that you don't need to collect. A white dishpan or similar tray in which to dispense the collection. I talked about this in the next video. Some forceps or tweezers or a fork, so you can move the material around, and if you have what you think is a positive detection, actually remove it from that collection. Some things to rinse your net and your collecting bucket, a flashlight or a headlamp, which is ideal to have out there in the early morning or late evening hours. Some way to document it, so a smartphone camera is just perfect to take a picture and to to report visually what you found. If you think you have a positive collection, a magnifying glass, something to write down where you are and when you're there, and a small jar with some type of preservative like rubbing alcohol or isopropyl alcohol. If you do come across a spiny water flea, it would be wonderful for us if you didn't only verbally and visually document that, but if you actually had the specimen and you had it on some preservation and you could share that with us, then we could make a definitive identification of it. So here's just some pictures of that material: a squirt bottle, a white sorting tray, and sort of dishpan thing that you have laying around in a closet or at the cabin, and some rubbing alcohol is just fine as preservative. There's a variety of different locations where you can source nets, but we have been in contact with a company called Aquatic Research Instruments, and I'll say a little bit more information about that company on the next slide. They're well aware of our program, and a number of people from our program last year and the previous year have reached out to them and ordered a net from them. So they're well aware of the type of net that is ideal for our surveillance monitoring, and they're ready to make that. There are other companies out there like Wildco that will manufacture nets that also can indeed be used. Virtually any net can be used for this if the mesh is small enough. It's just that some nets that, for example, nets that are traditionally used for veligers, zebra mussel veligers, might be in the business of clogging pretty more quickly than what we're calling here this 500 micron mesh net. So this is the company out in Idaho, Aquatic Research Instruments. They're out of Hope, Idaho. And they have this product, this spiny water flea plankton net. It's called that because we've been in contact with them, and we told them that's what we want them to build for our participants. The estimated cost we've just got an update this year on that. The cost of that went up a little bit to $370, but they can be contacted through online. I suppose you could call them as well. There's a newspaper article. They produce aquatic sampling instrumentation for a variety of different outlets, and they've been doing it for quite some time. So the effort. So what would be the effort if you had all the equipment and you say, yeah, I'm gonna go do it. I want to go out and participate. How much work is it actually going to take? What do I have to commit to? Well, one survey, what we're calling a survey of a lake at one time, is to go out to 3 different locations in your lake, preferably 3 locations that have some distance between them, and 3 locations that represent maybe 3 different basins of the lake or the deep spot in the lake and 2 flanking areas. The reason that we want to get offshore and in locations that are relatively deep is because, again, spiny water flea, they do retreat often during the daytime to the bottom of the lake, and they like it where it's dark, and it's dark and cold oftentimes out in the middle, deeper regions of the lake. And at each of those 3 locations, the effort is to do 3 tows from near the bottom to the surface, and to evaluate what you've collected at each of those 3 sites with 3 separate tows. So that's essentially 9 vertical tows, 3 tows at 3 different sites. And that completes a survey of a lake at a particular time. There is, this is sort of a little bit more detail on that, spraying down the outside of the net and checking for spinies. It can be done from a boat, but it can also be done from a canoe. So this is Meg Durr that was out there with a group a couple years ago, participated and did a survey, I think, on a lake up in the Grand Marais region. Again, so this is the telltale indicator morphologically for spiny water flea is this long caudal appendage. The entire animal is about a centimeter to 1.3 centimeters in length, and so that tail spine itself can push upwards of a centimeter, or about a third to 2/5 or so of an inch in length. There's really no other organism out there that has this long caudal appendage on it. There's some what we call lookalikes, so there are some other big organisms out there that might rival spiny water flea in total length. The one in the lower right-hand corner is called Chiabrus. It's actually a larval stage of an insect. Sometimes it's called a glassworm. Excuse me. This thing on the left is called Leptodora. The sort of the grayer, more transparent organism. They can reach up to 1.5 centimeters or so in length. And then there's this bizarre creature called Polyphemus. It looks every bit like spiny water flea without the tail spine. It's the closest relative of spiny water flea. It's native in North America. You probably won't find it offshore where you typically find spiny water flea, but they do exist in lakes. But aside from these 3 different species, there's really not a whole lot to get mixed up on. And again, size is a key difference. So here's an image of spiny water flea. Obviously, they're in the middle, and there's a couple native zooplankton on the right and upper right, things called Daphnia. So you can quickly appreciate the order of magnitude in weight difference between spiny water flea and most of what else you'd be gazing upon in a collection of plankton. Here's another image of it as well. So that's spiny water flea in the middle there. She's carrying a chamber of eggs on her backside, on her dorsal surface. And you see the black dots in there. She's got 4, probably maybe 5 offspring in there that are getting ready to hatch out. Okay, I'm going to transition over to Megan now, and she's going to chat a little bit about how to document a find.
Great, thanks, Don. Yeah, so if as you're doing your monitoring, you come across something that you suspect might be a spiny water flea, there's a couple of things for you to do. One is take a photo, or 2, your phone should do just fine with that. And you can give it a good check and make sure that it's not blurry. I know with tiny things, it's easy as you're getting closer for things to blur. So give it a good check, make sure it's a nice clear image. You can always let the spiny water flea be a small part of the image and it'll be sharp, and whoever is looking at it can zoom in as needed. You want to record the location within the lake that you found the spiny water flea, the depth of your sample, and the time of day. You will hopefully have a small jar along with you. I think Don mentioned that in the list of things to bring. So have that small jar and whatever kind of alcohol you happen to have, isopropyl or rubbing alcohol. Go ahead and bring that, put the spiny water flea in there, put it in the jar. I have some that I have preserved in hand sanitizer too. So if you're in a pinch, I guess there's options. Write the name of the lake, date, time, all of that information on a small piece of paper, and place that in or with the jar. An important point here is to write in pencil, not with ink. And then you're going to find your AIS specialist through Department of Natural Resources. That information is online. So you're going to report it directly to Department of Natural Resources, and you can do that either by calling them directly or by using a reporting tool called EdMaps. And then also report your results to us. And Don, if you can go to the next one, I think it has some more details on these. Here we go. So here's some of the info on the photos. So there's 2 ways to zoom on most phones. You can kind of do the pinch and slide, or there's sometimes buttons that'll say like 1x, 2x. The best thing you can do is to use the buttons so that the phone is switching to the appropriate lens and is doing a true optical zoom. If you do the kind of like finger pinching method, that can sometimes switch you to an optical zoom, which isn't a true zoom. It's just kind of like making an image bigger, but it doesn't change like the number of pixels and how sharp the final image is. So best is to make sure that you're using the various cameras, and your phone will know how to do that by using that 1x, 2x button that you see. So whatever highest level your phone allows for, it will be just fine. All right, next slide. Oh, there we go. Don't do that. And then here's the page on the Department of Natural Resources website where you can find who you should contact in your region. So if you go to the DNR invasive species page, you can see on that left-hand side towards the bottom, there's the contact an expert, and you can see aquatic invasive species. So you can see the map on their page, figure out who it is you need to reach out to, and their name and phone number will be on that page. All right, next one. Okay, and if you are wondering about which lake to sample, or you're interested in sampling more than one lake, or perhaps more than the one that you live on or recreate on the most, there is a tool called AIS Explorer that's been developed by the Research Center. And we will drop the link to that into chat here. But you can use that to get an idea of which lakes perhaps might be the riskier lakes. And Don, if you go forward one more, here's what the tool looks like once you get into it. And when you're looking at the top, you can see there's kind of some tabs. You want to make sure that you're on that prioritization for watercraft inspections tab. That's the one that has data relating to spiny water flea in it. So if you, on the left-hand slide, select spiny water flea as your species, and then where it says percentage of boats to inspect, slide it up to 100%, or then it'll give you a list of boats. So you can do it by county, and you can see which ones might be the highest priority lakes if you're looking for an idea on how to prioritize where you might go and do your sampling. All right, next slide. Oh, here we go. And then, sorry, one more slide about AIS Explorer. So you can also export that. So if you wanted to be able to print out a list or something, there's a way to export your data into a table. So you can view it that way as well. Okay, the next piece is the app, the reporting app, and I'm going to take over screen share and just kind of walk you through this live quick. So here we are. Okay, so this is the project's page for spiny surveillance. And there's this group of buttons right here. And if you click on the submit survey data, it's going to bring you to this reporting app. And it's been optimized that you can use it on a computer, or it's a nice view if you're doing it on your phone as well. And we've made some updates to it this year. So if you've done this with us before, it might look a little bit different. And the first thing you'll notice is that you're seeing a map as the first thing instead of the reporting form. This is here for you to view live where surveillance has happened. And that might be another way to help you prioritize your lakes or prevent duplication of effort. So we can zoom in here and kind of see all of these pink ones are previous years. So you can zoom in and see which lakes have had monitoring efforts on them. These green ones are lakes from us testing the app out. So those will disappear before we go live. But green ones are current year sampling events. So if it's a previous year, it'll be this kind of pinkish color. And if it's the greenish color, that'll be the current year. But you're going to want to go over to the submission form to submit your information in. You can put in your name, your email address, and we have a password in here to prevent spam from being entered in. The password is detect with a capital D. That's the same as last year. We're not changing it this year. You can pick what affiliation you're joining us from. From the drop-down, you're going to select county, and then you're going to find the name of the lake that you surveyed on. And for lakes where there's, for example, multiples of the same name, I'm not sure if I picked a county that has that or not. There's DOW numbers here that you can use to reference to make sure that you're picking the right lake. And if you're not sure about the DOW number, you can click right here to Lake Finder to find that information. But I'm just going to pick one of these and say it was today. The time, we'll just enter a time in, and you can enter in the depth, and if that's feet or meters, how many sites you sampled, how many tows you completed at each site, We're asking about how long it took you to do this. And then just some information about your net as well. So hopefully you have this information, and for anything you don't have, you can just put a note in that says why it's not there, or if you have questions about it, there's a space to enter notes. Next one is if you found something or not. So there's no, or a yes, I think I did, or maybe I'm really not sure. Both of those give you options to upload files in. You can just select here and add files. And then you have a spot to say anything else. Once you're done, you submit your form, and then we can see our update down here that it's been submitted, and refresh the page if we need to submit something else. So that's the reporting form for this year. Not a lot of changes if you did this last year, but a little bit different. And I'll let Don start the slides back up. One more forward.
And while Don's doing that, can I just emphasize, we really need those zeros. Remember, Don said that we can't tell the difference between nobody sampling it and somebody sampling it and saying nothing was found unless we get these reports. So zeros are actual data. And so that's what we're looking for. So please, please, please do this. Fill this out every time you go out and check a lake.
Yes, thank you, Val. Agreed. And also helpful, even if you're submitting zeros, that now it adds you to a map, so other people can see that lake's already been surveyed. I don't know if there's anything else to add with this slide as well, so we can go to the next one. I think we got that all covered with a live demo. This was the results over the last 2 years. We've had 21 different people so far who've taken part in this, 42 different lakes that have been monitored, 89 different surveys total. And as Don mentioned before, no spiny water flea detected so far from this. And that's a number that we hope to continue into 2024. All right, next slide.
I'm trying. Okay, here we go.
Here we go. All right, Don's going to take over again.
Okay, here we are. Yes. Okay, frequently asked questions. Don, can this be done from a canoe or kayak? Yes, it can. It's obviously a boat is a more stable, just easier platform to work from with all the steps involved. But if you have experience and you can do it safely, absolutely a canoe, a kayak as well if you have some space to operate. I've done myself plenty of surveys in backcountry areas from at least a canoe, and it's been easily done. So will our zebra mussel veliger net work? Yeah, and I sort of alluded to this earlier. Any net will work if the mesh size is small enough. The only problem with the veliger mussel net is that because the openings are so small, those nets are probably going to be likely to catch a variety of different plankton that is going to make your scan of the water less efficient to begin with. But the real challenge then is going to be when you dispense that into a tray to do some identifications, and especially maybe in late August, if there's an algal bloom, for example, in your lake, you're going to be pawing through all sorts of phytoplankton that might obscure your ability to detect some of the zooplankton in there, even spiny water flea, despite their size. But if that's what you have and that's what you want to, that's what you can use, go for it. I think it's absolutely appropriate, and because the reporting form asks you to identify what net you use, that will help us understand as well what that effort might have looked like compared to an effort where you had a bigger net with larger apertures. So how do I find the deepest spots in the lake if I don't have a depth finder? Lake Finder, which is that portion of a website maintained by Minnesota Department of Natural Resources. Through Lake Finder, boy, they have information on the vast majority, I believe, of Minnesota's lakes. And for many of those, there are maps, bathymetric maps that can be downloaded. And through that, one can identify best locations to go out and sample deep locations. And it's nice to know as well, right? Because when you get to a site, you want to know, roughly speaking, how deep it is, especially if you don't have a depth finder. So you do two things. One, you sample most of the water column, and two, you don't deploy your net all the way to the bottom and start off by bringing to the surface a whole bunch of mud that is going to take some time to clean and deal with. What kind of weather conditions are best for spiny surveillance? Calm conditions are fine. Calm conditions are great. They don't behave one way or another any different when it's windy versus when it's calm. So you don't have to go out in the wind. The problem with going out in the wind also, as I tried to indicate in that video, is it's going to push you around if you don't anchor, and that's going to prevent your net from going all the way to the bottom before you begin your retrieve. And because spiny waterfly is pretty good in the daytime at descending to the bottom, near the bottom of a lake, they might be there and you might not be able to capture them. So weather conditions, honestly, the calmer, the better, just fine. If you can do it in the early morning or the later evening, that's good as well, as long as time and safety are prioritized there. But honestly, in lakes that are established, we can go out, especially stained lakes. So Island Lake Reservoir, where I filmed those videos, is a stained lake, tannin stained, very, very brown water. And animals there are swimming throughout the water column day and night. It's more these very clear water lakes where they tend to descend deeper in the water column during the day. Okay, are 3 plankton tows at 3 sites really necessary? Well, any effort is good effort. That is what I would say. The reason why we've gone with 3 plankton tows at 3 sites is there was a published study out of Canada that demonstrated even in low density Bythotrephes lakes, an effort of that magnitude has a very, very high chance of collecting at least a single spiny waterfly, making a positive detection. And so that's why we identified that as what we consider sort of as the baseline effort to make a call on whether or not the effort was thorough enough to know that they weren't there, to say that they aren't there if they're not there, sort of a thing, or to detect them if they are there. But if you can go out and do 2 sites and 2 tows, do it, right? So any effort is good effort, and we'll know what that effort is once you report it. We have, I think this is our last slide, sort of identifies resources for the volunteers. There's a web page there. There's a variety of, as we've indicated several times, resources there to take a look at, instructional videos. Val Brady and Meg Durr and I put together a manual, like 13, 14, 15 pages manual with all sorts of pictures in there that sort of is the unabridged version of this 1 hour webinar. And then there's email and phone in there as well. I'm sure my email and Val's email is in there, and well as MAISRC, and I'm more than happy. I've responded to a variety of different contacts, people that are even out in the field and will email me with something. If I'm around, I'm happy to respond to something like that. And it's exciting. I would encourage you to try to get involved with this. It would just be fantastic to continue to not only maintain but to grow our effort of surveillance in Minnesota. So thanks, everybody.
Thanks, Don. I will add too that if you do participate this year, we would love it if you took photos of yourself or had someone take a picture of you out in action and sent them our way. We love to be able to share them, and they become parts of training materials and slideshows like this one. So we would love that. All right, I'm going to take over the screen one more time. We have the Q&A feature up. Here's again how you access it. You can upvote questions as needed. We have just a little bit of time left, and I think that's okay because there's about 3 questions so far, but feel free to add more in as needed. So for Don and Val, the first question was wondering if spiny water flea can survive in rivers or in like the lakes or impoundments on those rivers, like Lake Pepin was given as an example.
Yeah, they probably can for some amount of time. We don't think of them as lodics or sort of riverine species. They're not strong swimmers, in fact, and so they're gonna get washed with the current. They're not like sort of aquatic insects. They don't go to the bottom and hide underneath rocks, and so they're gonna be transported. But if there's plankton there, and they're not getting damaged by the current, sure, they can survive there for some amount of time. In most natural lakes that support populations have a deep, dark environment that they can retreat to during the day. So rivers oftentimes, except for deep pools in certain areas, aren't slow and deep enough to really offer the type of habitat, the type of lake habitat. So those would not be ideal locations to find an existing population, but certainly they serve as corridors through which they can move from one lake to another. So the idea of sort of hanging a net in flowing water and hanging out for a while and waiting for it to fill up, sort of an intriguing way to think about how they might move from an invaded lake to an uninvaded lake. But otherwise, if you're going to put some effort in, I would go to a lake and not a river. Yep.
Great, thank you.
Mhm.
Okay, next question was about, oops, I just cleared it out, but I remember it. The next question was about, you had shown a graph of the abundance, I think, during different months of the year, and I think June had nothing.
Yeah.
So the question was there no data collected then, or was that you could not find spiny water flea in June?
Yeah, that's a really good question. It was that they weren't available in the water column to be collected then. They were still in the resting egg stage, or they were just beginning to emerge from the sediments from those overwintering eggs. And so it takes temperature and it takes food for their populations really to get going. And at this latitude in northern Minnesota, that was from Island Lake Reservoir, it's really not until mid or late June until densities start to show up in detectable numbers in the water column. So it takes some time. More southern lakes that warm up faster, like Mille Lacs, for example, those populations are probably detectable in early June. But so you could kind of see how fast the population in the water went from undetectable to 5, 20, 40 individuals per cubic meter, right? In a relatively large lake. So yeah, it wasn't lack of effort. That we sampled that lake every week for 3 years straight. So we knew when they were there and when they weren't.
Great. All right. I see. I'm gonna elevate one of these up to the top because there's one that's wondering about how to clean a net if, for instance, you wanted to visit more than one lake.
Ooh, yeah, that's a really good question, because the last thing that we want to do is be part of the problem here. And so thoroughly clean it, thoroughly rinse it, visually inspect it, turn it inside out, pull off every bit and piece that looks like spiny water flea that you can, and then let that thing dry. Let it thoroughly dry. At minimum, I would let it dry overnight. There's recommendations from the DNR to go for 5 days, but overnight is, if it's really dry, is probably going to be sufficient to kill all the spiny water flea. We know that the eggs can survive for 4 hours. The resting eggs can survive for 4 hours under dry conditions, and the spined animal that you see sort of resting on my finger or someone's fingertip there, their heart can keep beating for up to an hour and 15 minutes if you take them out of water. So we haven't published that work yet, but we've got a lot of documentation that shows that they can survive for at least an hour out of water, even if their surrounding environment is dry. So drying is the best way to go. You can pressure wash a net as well. The great thing about drying is it's the easiest, it's the cheapest, it's the less destructive. There's some chemicals that you could use as well, but I would go with drying, and if you just if you can't do it, if you're going to do 2 lakes, go to 2 different lakes in one day, pick one of them and do the survey there, to not risk moving them to the next lake. And you move everything around, right? I mean, you're not just moving spiny water flea around. Anything that's in Lake A, you go to Lake B and you don't clean your net, you just hitchhiked all of it. So interesting to think about that. Yeah.
Wonderful. Okay, I see it's 4, but if it's okay with you two, there's one question left, so I might let us run just a little bit over.
Oh yeah, I can hang around.
All right. So next question was if there's a maximum depth. So for example, the lake that this person would go to has a maximum depth of 110 feet.
Mm-hmm. Yeah, that's a deep one. Well, so you're going to need a net that's 108 feet long. No, you can, yeah, sure. If you can get all the way to the bottom or close to the bottom, that's ideal. You know, with the deployment, you really don't want to smack the bottom with your net. So if you stay 5-6 feet off the bottom, that's fine. If you're, you know, if you don't want to take the time, if you just don't have the capacity to deploy the net all the way to the bottom, think about going beyond the thermocline, right? So in the summer, you know, warm water on top, and then the big transition to cold water. And that transition is the thermocline. If you can get down below that, and that's usually somewhere in the top third of the water column, is where that sets up. You're likely to be sampling in the region of the water column where they are during the daytime. But the deeper you can go, the better, for sure. You might be better off if you have a spot that's 110 feet deep, and you can't get there. You can get to 60 or 70 feet. Go to a location that's 60 or 70 feet deep in the lake and sample all the way to the bottom there. So you'll get the full water column in what's still a relatively deep part of the lake. Yeah.
Great.
Yeah.
Okay. Val, do you have anything that you want to add before we wrap up?
So just a reminder, somebody said this in the chat, but just make sure everybody saw it. If you do find something suspicious and you're preserving it in something alcohol-based, you need to be sure that you're writing with a pencil. So make sure you have a pencil along. Even Sharpie won't necessarily survive a dunk into the ethanol. And then, you know, you might remember most of the stuff, but it's really a good idea to just have a pencil along. And a pencil might also write when things get wet. So just a tip there. And I can't overemphasize the cleaning, because the last thing we want to do is move things around. But you've got to think about not just the net, but all the other gear that have contacted the water that you're taking with you. And so that's your boat or your canoe, and whatever other gear you used in that first lake before you go to the second lake. So clean, drain, dry. That's the message.
Great.
Well said.
Well, thank you to both of you for being available for this training webinar. The last couple of items that I have are to watch for the recording. So if there's anything you wanted to review or you need to share with someone who couldn't make it, that recording will come out. We will send an email notice when it's available, and we will post it online. We'll also be sending out little reminders and messages now throughout the sampling season, reminders of when it's time to like have your gear ready, or if you haven't ordered your net, you probably should have by now, or now it's peak sampling time, get on out there. So watch for some messages from now through September-ish. And if you have any lingering questions, please feel free to reach out to us. You can email us at aisdetectors@umn.edu, and Michelle and I will help. And if there's anything that we need to pass on to Don and Val, we'll be sure to get that to them as well.
And don't forget, Don made those really nice how-to training videos. They're on the website. There's 4 of them. Each one's about 5 minutes long. And so those are really good to review. You can even, a lot of people like to do this with their grandkids. You can show the grandkids so they're ready to help you when you get out there in the boat.
Yes. Great. Well, thank you everyone for joining us, and we're excited to hear about your spiny surveillance adventures this summer and fall.
Very much. Thanks, everyone.